Assalamualaikum and hi, my name is Siti Nurzaira binti Kamaruddin. So today we're gonna learn about diversity of marine phototroph. Before I move further into this chapter, so I would like to elaborate more about the subtopics. There are several subtopics that are to be learned in this chapter. So I would like to continue first with the introduction. Okay, so what is meaning of phototrophs? The word origin of phototroph is coming from two words, which is photo and trophs. The photo means light, while the troph means nourishment. Okay, so the full meaning of phototrophs is organisms that use energy from sunlight to synthesize organic compound to use in a metabolic process. Phototroph have two types, which is photoautotroph and photoheterotroph. What is different, which is these two kind of things? Okay, the main difference of photoautotroph and photoheterotroph is photoautotroph can synthesize their own food, while photoheterotroph solely depends on the light energy. Now, let's move with the first subtopic, which is traditional understanding of primary production. In 1970, the atoms and flagellates were found as the most organisms that contribute into the primary production, as they are the major organisms that can be collected in the planktons. These include in eutrophic and ergotrophic environments. There are two reasons. They were easily speculated and the method detection at that time. The plankton have three types of mesh size, which is 100 micrometer, 70 micrometer, and 60 micrometer. And the results show that the phytoplankton that have colony size more than 60 micrometer can be used in the research. In this recent research using DNA detection and flow cytometry show that the predominant phytoplankton is much smaller than those plankton that have been collected. For clear example is haptophytes and coccolithophore. Now, we move to the second subtopic, which is recognition Pico Cyanobacteria dominance. The technique that have been used is fluorescent microscopy. From electron microscopy observation, it revealed that unicellular Cyanobacteria Synechoccus species are predominantly distributed in tropical and subtropical regions. They are very tiny cells with range 0.5 until 2.0 micrometer in length and 0.5 until 1.0 micrometer for diameter. They also play significant role as a primary producer in that time. Why? In the late 1980s, the development of technique is increasing and they have used HPLC technique. This technique is used for pigment analysis and cell detection technique by using flow cytometry. By using this technique, they have revealed a new Synechoccus species named Prochlorococcus, and this species dominates in subtropical and tropical regions between latitude of 30 degrees north and 13 degrees south. They have much smaller size. The range size for diameter is 0.4 until 0.6 micrometer, and for the length 0.5 until 0.8 micrometer. This species contain divinal chlorophyll A, which had an absorption spectrum slightly different from that usual chlorophyll A. This species did not contain picobelly pigment. And lastly, this species also played main role as a primary producer in that time. Now, we move to the third subtopic, which is discovery of ubiquitous photoheterotroph. In this subtopic, there are two main things that need to be known, which are wide distribution of AAPB contain PHCl and proteorhodopsin contain bacteria. Now, let's move to the AAPB first. APB was reported in 1979. AAPB was derived from non-sulfur purple bacteria and widely distributed in aerobic environment. They also need oxygen for photosynthesis process. And AAPB distribute widely in marine environment. The in situ growth rate of AAPB was faster than the other heterotroph bacteria. Now, let's move to the ANAAPB. ANAAPB can be easily found in a proteobacteria. ANAAPB does not need oxygen for photosynthesis. ANAAPB distribute widely in anaerobic environment. The amount of primary production of ANAAPB reach up to 30% of the total primary production in same specific lakes. Now, let's move to the proteorhodopsin. What is proteorhodopsin? Proteorhodopsin is the rhodopsin-like protein detected in marine proteobacteria. It is a radial opsin in complex in a member of rhodopsin. Rhodopsin is well known as a photoreceptor in vertebrates as a light sensor. Let's move to the new subtopic, which is oxygenated photosynthesis using far-red light. Okay, first we go to the chlorophyll A. Chlorophyll A is indispensable for oxygenated photosynthesis since it's played role in light harvesting and others. The quantification amount of oxygenated phototroph and the activity of oxygenated photosynthesis is based on chlorophyll A. The range of in situ light absorption by chlorophyll A is from 400 to 700 nanometer, and it can be called as PAR or PAR. The function of PAR is to evaluate of light strengths for the activity of oxygenated photosynthesis. Next, we move to the chlorophyll D. Chlorophyll D was reported in 1996. Chlorophyll D is a red shifted chlorophyll which can absorb far-red light 30 nanometer longer than chlorophyll A. Cyanobacteria employs chlorophyll D for the chlorophyll for the reaction center. As a result, Cyanobacteria can perform oxygenated photosynthesis using far-red light 700 to 740 nanometer, which cannot effective to other phototrophs. Acariochlorid species. Acariochlorid species is a unique organism as it can perform oxygenated photosynthesis using chlorophyll D. Acariochlorid species widely distributed in coastal area as epiphyte on seaweed. The amount of chlorophyll D on seaweed range between 1 until 13% versus total chlorophyll A contained in seaweed. The energy transfer efficiency of photosynthesis using chlorophyll D is equivalent to that using chlorophyll A. Next, we move to the chlorophyll F. Chlorophyll F was reported in 2010. It was reported from a Cyanobacterium isolated from the microbiome on the stromatolite at the coast of Australia. The maximum absorption of chlorophyll F can absorb around 720 nanometer, but chlorophyll F can absorb until 760 nanometer. The chlorophyll F was not taken as primary producer in marine environments. These findings on far-red utilization for oxygenated photosynthesis required reconsideration of the PAR concept and primary production in marine environments. Now, we continue with subtopic which is discovery Pico phytoplankton. In recent years, very unlike Pico phytoplankton species have been revealed to be widely distributed. However, until the early 2000s, only some dozens of species in Chlorophyta, Heterocotyphyta, and Heptophyta were known as eukaryotic plankton. Around 2010, there are analyses of phytoplankton using metagenome and flow cytometry. By pigment composition analysis of marine water, a carotenoid 19HF was detected to be at the high latitude areas from 40 degrees to 60 degrees of both north and south. A similar estimate was reported. Picoheptophytes form 5% of the global picophytoplankton biomass and contribute significantly to the primary production. In 2007, picophytoplankton, which did not belong to any known phyla of algae, were discovered by metagenic analysis. The DNA was clothed with the cryptophysian lineage, and it was tentatively named as cobblephytes. The picobellephyte distribute widely in coastal area of Europe and the North Atlantic. The cell number was reached up to 1.6% of the total eukaryotic phytoplankton. In the 2011, a new candidate phylum was detected by metagenomic analysis targeting plastic DNA. Monarchs, the name tentatively given. Monarchs widely distribute in the coastal area of Europe's Atlantic and Pacific Ocean. Since algae in both candidate phyla have not been isolated, all the details about characteristics are not yet known. Now, we move to the sixth subtopic, which is strange phototropic microorganisms. In this subtopic, it emphasizes about the unique microorganisms that have particle UCYNA. The cyanobacterium, like a particle UCYNA, was detected by using metagenomic analysis of nitrogen-fixing bacteria in marine waters. It was first detected as a cyanobacteria that have nitrogenous gene for a nitrogen fixation bacteria in pelagic waters. This particle also contribute into nitrogen cycle in pelagic waters. Usual cyanobacteria grows by an oxygen photosynthesis process photosystem, respiration system, and a gene for carbon fixation system. But however, the UCYNA particle lacks of many things. It lacks TCA for respiration, rubisco, photosystem 2, and enzyme for fixing CO2. Because of this lack, they have suggested UCYNA might be symbiont in other organisms, since it possesses a complete set gene for photosystem 1 and contain chlorophyll. It might use light energy in other activities of the particle. Okay, now we move to the last subtopic in the diversity of marine phototrophs. In this subtopic, there are few terms that are need to be known, which is first, photosynthesis. Photosynthesis is a classically reaction that can reduce carbon dioxide into organic compounds such as sugars using sunlight as a source energy. Next, primary producer. Primary producers are organisms that synthesize organic compounds using inorganic carbon as their carbon source. They are representative of photosynthetic organisms and chemoautotrophs. Third, primary producer. Primary producer have been used synonymously with photosynthetic organism. Similarly, it has been considered phototroph and photosynthetic organisms are almost the same so far. However, recent discoveries of new marine microbes suggest that the term phototrophs, photosynthetic organisms, and primary producer need to be changed. So, the new meaning of phototroph is an organism which can synthesize ATP using light as its energy source. So, here it is, the extra info regarding the diversity of marine phototrophs. The color plate showing the graphs of pigment and bacteria rates. The A graph show the pigment analysis and retrieval of phototrophs, and the B graph show the general bacteria library analysis with refraction and abundance data. The image at the right side is an article about the diversity of phototrophic bacteria in microbiomes from the Arctic hot spring. And at the left side is phototroph phylogeny. Several phylogenetic landmarks in the evolution of photosynthetic organisms are highlights on the tree.